There are really two currents in medicine, I would argue, but in particular in cancer medicine, that make this, for me, one of the most fascinating times in certainly my professional career, the most fascinating. When I was a fellow in Baltimore in the mid-'80s, as a medical oncologist, we were trained very simply to look at patients, especially patients with advanced disease, in the context of the organ site of origin. So as we all know, those of us who are physicians in the room, everyone with colon cancer treated a certain way. In that day and era, it was 5FU in many different ways. Or everyone with breast cancer in another set of ways, lung cancer likewise. And obviously with the Human Genome Project, with the Cancer Genome Atlas, with the revolution and the understanding of tumor molecular biology, we now recognize that just as each of us is a unique individual, when we have the misfortune of developing a cancer, that cancer is unique. And it basically, as we say to lay audiences, it has a unique biological fingerprint. And we now realize, without hyperbole, that sometimes one person's colon cancer can biologically be more like someone else's lung cancer or yet another person's breast cancer. So Jens was making that point, and as far as I'm concerned, he's absolutely correct. The other trend, which to me is even more exciting, is that for our children and grandchildren, the concept of illness is going to be redefined. And what do I mean by that? In cancer, we're often accused of reacting once the proverbial horse is out of the barn. The tumors are, even small tumors, are relatively well advanced. And with the understanding in tumor biology that I've referenced, and the human genome in general, and to include germline implications, we now are understanding how to identify individuals and populations at risk. So in essence, a generation or two from now, illness will sometimes be seen as the risk for what we now call illness, if that makes sense. And our effort at the Cancer Institute is more and more shifting to managing populations that are defined as at particular risk or individuals at particular risk. So those two trends are key. Now, one of the themes of today's lecture was teamwork, and I want to tell you very quickly in the remaining minutes about the personalized medicine program that has grown up here as a true team effort, a multidisciplinary effort, a multi-professional effort. Back in 2014, we began our strategic plan, and we focused on this concept of the unique biological characteristics of everyone and each person's tumor. But we also realized there's another component to personalized medicine, and that is caring for the whole patient, to include addressing family, socioeconomic, psychological, environmental, and supportive care needs. We have a very robust set of supportive care services, survivorship, palliative care, psycho-oncology, cancer rehabilitation medicine, genetic counseling. I could go on. We have 18 of these services. For today's purposes, I'm going to focus on the second aspect of personalized medicine, that is the unique biological characteristics of each person and each person's tumor. So this is a brief history of what we've done and as a team. And the importance is that we probably now have, with everyone's help, to include folks who reside within SNI, we've assembled what has been noted at the national and international level as a very comprehensive and integrated program in tumor genomics with a clinical application. So back in 2014, we made some critical decisions. The first was, okay, are we simply going to send tumors out to Foundation Medicine or Caris MPI or whomever, or are we going to do in-house sequencing? We decided to do the latter because we wanted that interaction between the pathologist, the molecular pathologist, and our clinicians, our medical oncologist in particular, but also our surgical specialist. We started by recruiting Dr. Anna Berry, who was the head of molecular pathology at UCSF, and Anna and Dan Ben-Shu, who came out of Mary Claire King's lab, the BRCA lab at the University of Washington, helped us assemble our targeted sequencing panel. This is an evaluation that's done on paraffin-embedded tissues. And we started small. We started with a 68 gene alteration panel that was focused on the most actionable tumor alterations. So these are mutations in DNA, the most actionable tumor alterations as based on the existing evidence in 2014. The other point is we encouraged our doctors to do this profiling as early in the course of treatment as possible, that is, at the time of diagnosis, if at all possible. Mind you, I'm a phase 1 investigator by training and profession, and we tend to use biologic profiling in phase 1 work to discern the therapy of last resort after folks have exhausted standard therapies. Again, in this day and age, we're trying to move that up so that it's part of the initial pathologic interpretation. The second point, in the fall of 2014, realizing that this field is uncertain and that there's a lot of controversy as to how to use molecular profiling in the management of cancer patients, we said, well, we can't dictate what our physicians will do, but we can formally follow what they're doing. So we started an IRB-approved registry protocol to basically evaluate how physicians, our medical oncologists in particular, were using this data to make decisions either to do something or not to do something. Sometimes this profiling tells you what not to do, which is just as important. To date, we have, as of a week ago, we had 1,009 patients on our registry. We hope to increase that rate of accrual in the near future. I won't go into the details of all of that. The third step in January of 2015, and those of you who are here at Swedish Cherry Hill and First Hill, I would invite you anytime that you want to participate in our molecular tumor board. This is now one of the older continuously functioning molecular tumor boards in the country. It meets every 2 weeks, Monday morning. And effectively, we do what you do here, what you were doing a few minutes ago, but focus specifically on the genomic alterations in a patient's tumor and how that might inform the overall management of the patient. In late 2015, we became one of the early adopters of an IT platform called SciAps, for the neurosurgeons, Synapse without the N. This is a startup out of the Bay Area. It's a cloud-based platform that effectively bolts onto Epic and helps us collect, organize, and analyze the genomic data in the context of the clinical information in Epic. And I'll show you what that looks like. Lastly, in the spring of 2016, we opened our early phase clinical trials unit, the Reed Family Innovative Therapeutics unit, on the 4th floor of the Arnold Pavilion. And this is basically a 21st century phase 1 unit that's focused on developing new agents, new anti-cancer agents, to include the exciting biologicals, in the context of the profiling that we're doing. So these agents being informed by the genomic profiling that we're increasingly doing on a routine basis. So that's the basic history. This is probably one of the more important slides because it shows an integrated IT platform that we've been in a very stepwise fashion developing over the past 3 to 4 years. In the center, you can see SciAps. That's a cloud-based platform that bolts onto Epic. By the way, through our enterprise data warehouse, because we have various instances of Epic throughout the larger system. What's unique about this platform, it's connected to the sequencing labs. In our case, Cellnetics, our partner in this effort. It's also connected to our AP laboratories, also in our case, Cellnetics. It's connected to our clinical trials management system for medical oncologists. The CTMS is critical. That's the IT platform that supports our clinical trials effort. I think the neurosurgeons in the audience know that in medical oncology, to practice state-of-the-art medical oncology, you need to be on a daily basis engaged with clinical trials because of the evolution of medical oncology, the practice of medical oncology and new agent development. This platform also includes, importantly, a connection to our tumor registry, the tumor registry, which is the cleanest data, though there's a lag time, as we all know, in that data being scrubbed. We also have connections to disease site registries. We have one of the larger breast cancer registries in the world here, over 17,000 patients. And that also connects to this. And then importantly, we have connections to state national registries to include an evolving connection to the SEER database, the CDC-sponsored epidemiologic database that in the Pacific Northwest arguably is one of the better SEER platforms in the country. So this platform has received a lot of recognition, again, for its comprehensiveness and its integration. We're now in the process of adding on machine learning capability to this platform. Now this, in some ways, those in the room who are of a younger generation, this is what you're going to be doing routinely at your office desk or in the clinic office. This basically allows you to sit and look at the patient in front of you and basically compare that patient in terms of their clinical demographics and genomic profile to patients who exist in either our database or associated databases. So increasingly, I'm a clinical trialist. That's how I was trained in Baltimore, and I dedicated my life to early phase clinical investigation. But some clinical trial work is increasingly being replaced by real world evidence. I think many of you know that term, and part of real world evidence is mining data systems like this. So an important point. Now, I just want to briefly show you what our molecular tumor board is like for those of you who haven't participated. And this is an actual case from the spring of this year. I won't go through the history, but we present the history of the patient. In this case, a patient with heavily pretreated ovarian carcinoma who had received standard chemotherapy. We do show light microscopy. That was something that I insisted on. At present, we do not show radiographic data, although we may start doing that. Now, this is the key. So what do we have here to the left? This pointer doesn't appear to be working, but to the left where it says aberrations, those are the two gene alterations found in this patient's particular tumor. Now, what I like to say is that for younger medical oncologists, I remember in the early 80s when CAT scanning was first coming into real usage, and we were marveled by being able to see within the body at scales of 2-3 millimeters and then 1 millimeter. It was fascinating, wasn't it, for those of you who were around then? I would submit that for the younger medical oncologists who are beginning to practice, these mechanistic pathways, what I have to the side is the relevant mechanistic pathway that shows where this lesion resides and effectively the impact of the gene alteration. So for the next generation, the CAT scan of the 21st century is this kind of mechanistic pathway. That's a concept that I think is worth contemplation. So we review the relevant mechanisms of action and how they're perturbed. And then we review the possible therapies, either on-label, that is FDA approved, or off-label. If a drug, I think most of you know, if a drug is approved by the FDA for one indication, but there's evidence, especially in the medical literature, for its use in an unapproved indication, that's considered standard of care, certainly in medical oncology. So on and off-label usage. But importantly to us, possible clinical trials. I didn't mention that IT platform has as part of its architecture a clinical trials matching function. So we have an automated clinical trials matching function that refreshes on a daily basis, and daily basis in terms of the available data networks, both locally and nationally. We're actually in the process of revising this with SciAps to take it to a more sophisticated level. I should mention in red, you see that's taper match. That's the ASCO-approved basket trial where patients are being assigned to off-label drug usage based on their gene alteration and not their tumor site of origin. And again, that speaks to the initial points that both Jens and I were making. Now, this slide is just to say, well, to the uninformed, they look at what I just reviewed and say, great, you have a patient, you have their tumor, you have a gene alteration, you have a drug match. This is wonderful. Well, we're very early in the field. As of the spring of this year, this may be slightly out of date, there were only 22 drugs approved by the FDA with an associated gene alteration as a biomarker. So we're very early in this field in both understanding the range of mutations that might be actionable, and of course, as new agents are developed, there are more relevant drugs. This slide is emphasizing the program here at Swedish at the SCI has informed the rollout of a genomics initiative across the entire Providence St. Joseph Health enterprise. And mind you, it's not just about targeted sequencing. Soon we're going to be doing whole genome sequencing, or at least whole exome sequencing, some degree of transcriptomics, but probably most importantly, proteomics. The realm of proteomics, which is obviously in some ways the gold standard for evaluating how a gene alteration affects the human body, is really the next frontier, in addition to immune profiling and maybe some more arcane areas like microbiomics, which is looking at the gut flora and understanding how the gut flora impact on overall health and tumor biology. Now, I want to finish by showing you a few quick examples of what we've done with our database. We've reported out at a number of national and international meetings in the past couple years. This is a presentation that we gave at AACR, the American Association for Cancer Research, earlier this year, quite simply showing that in 20% of our patients who underwent profiling or in this registry, the doctors felt that the genomic data immediately impacted their decision making. And mind you that sometimes it's not what you do, but what you don't do. I'm a GI oncologist, and in colon cancer therapy now, it's well recognized that if you have a tumor that's KRAS mutated, then you don't use EGFR-directed therapies, period. And in fact, they won't be approved by third-party payers. Now, this is a bit of work that Tara Benkers led that we're very proud of. This was reported at ASCO, the American Society of Clinical Oncology, 2 years ago. And as we say in the South, you all have a very large neuro-oncology practice. And this was, at the time, we believe, the largest encyclopedic report of molecular profiling in primary brain tumors. So at the time, this was interesting and important information, and just underscored that there are potentially a number of actionable targets that tend to occur in primary brain tumors with relative frequency. That was the take-home message of this work. And again, this was on almost 100 patients with primary brain tumors. Now, this is another bit of work that's quite interesting. Many of you may know Chuck Drescher, who's one of our GYN oncologists. And Chuck led some work that was ultimately presented in Munich at the EORTC meeting that focused on looking at P53 mutations. P53, as some of you may know, is a very common mutation. It's ubiquitous. And at present, it's really not actionable. P53 is a tumor suppressor gene. So it's the brake, right? And when it's mutated, the brake is released. So you get abnormal cellular growth. Sometimes, though, in addition to the brake release, there's a gain of function. And this is based mostly on laboratory investigations, in vitro, in vivo. And we wanted to interrogate, well, how many of our patients appear to have these GOF, these gain of function mutations? It turns out that it was 10%. And interestingly, these occurred throughout the different organ site diagnoses, with the exception of breast cancer. So this is the kind of data mining that leads to hypothesis-driven work. We now have to understand, well, what is the significance of these gain-of-function mutations, and do they really not occur in certain tumor types like breast cancer? This is another piece of work that we're doing with colleagues that you all work with on an occasional basis, the Institute for Systems Biology. This is not about tumors, but it's about trying to understand the biological underpinnings of common side effects of chemotherapy. So Lee Hood, Nathan Price, and I wanted to take the concepts of scientific wellness, for those of you who are familiar with that approach, and apply it to cancer survivors. So what we're doing here is evaluating 100 women undergoing chemotherapy for early stage breast cancer, doing biologic profiling with whole genome sequencing, proteomics, metabolomics, functional MRIs. Bart Keogh is a collaborator in this project, by the way. We're looking at this data before, during, and after chemotherapy and trying to find biomarkers for common side effects of chemotherapy. And if we understand those biomarkers, the underlying mechanisms, maybe we can develop interventions to reduce the side effects of chemotherapy, things like peripheral neuropathy, or importantly, neurocognitive changes, so-called chemobrain. If we can understand the biological underpinnings of that phenomenon, we can hopefully ameliorate it or even eliminate it altogether. Now, the last example I want to give is in some ways the big idea. And when we began developing our program, we began using this platform, SciAps. We began working with Stanford and Intermountain Health because they also were early adopters of this particular platform. Jim Ford, Lincoln Nadal, and I got together and we said, why don't we share de-identified data? This attracted the attention of Vice President Biden's Moonshot Initiative, and we were invited back, boy, it seems like ages ago, doesn't it? About a year and a half ago, to participate in that event based on this data sharing, because part of the Moonshot Initiative was to work across traditional barriers to share big data. So the idea here is very simple, to share de-identified data that relates to genomics as well as basic clinical and demographic information, and to use this information both in clinical application as well as in research. We have a number of institutes and systems that are about to join us. The Henry Ford system is going to be joining us in this effort within the next week or two. We really are just turning the switch on this platform. We had our first inaugural meeting just 2 weeks ago of the founding group. But as you can see, we have a number of important health facilities around the country that are in the queue. And this soon will be one of the larger, if not the largest, data sharing platform in tumor genomics with literally hundreds of thousands of patients. And this is really the future. This is part of that real world evidence that I alluded to. Likewise, because of the platform that we've developed both here and increasingly in Portland and throughout the rest of Providence St. Joseph Health, we've been invited by the American Association for Cancer Research sponsored Geni project to participate in their data sharing platform. It's a little more research in orientation. We're actually going through, we were invited to apply and we're waiting for the final decision, but we anticipate participating in this effort as well. This is what I alluded to, which is increasingly we're going to take these large data platforms and do what with them? We're going to create machine learning platforms. So we have a project with GNS Healthcare out of Boston to basically take the group of breast cancer patients that are in our breast cancer registry, but also in the genomics registry, to use that data to model a platform to help us in decision-making at our molecular tumor board. It's a proof of principle project, and if this works well, we'll be doing it in other disease types. For example, in neuro-oncology would be a terrific place to do so. We believe in creative collaborations. That's what we're all about. And finally, as a drug developer, I believe that routinely, not only in standard of care, but in assigning patients to clinical trials, we're going to be using this broader biologic profiling to maximize the chances of patients benefiting. So this indeed is the last slide, and it emphasizes the challenges that we have in ascending order, in my humble opinion. And the greatest challenge is expanding our comfort zone, because this is indeed a new era. So I want to thank and recognize our team. Phil Gold is my co-PI on the Genomics Initiative here at SCI and the Deputy Director for Research at the SCI. Anna Berry is our Scientific Director. Chuck Drescher, GYN oncologist, is an important co-PI. He's actually the PI on the ovarian spore grant that's being reapplied for at the Hutch. John Pagel is the head of our emerging and very important heme malignancies program. And I mentioned Dan Bin Shu, who's our technical expert at Cellnetics. Thank you.